This is the new title here. So my apologies, and I'm really sorry not being able to be in Rome with you guys. You know that Rome is my second house, and some people in Rome, including Andrea, are like my family. So it's really a pity that I cannot be there with you sharing this this occasion, coming back to life and to almost normal activity slightly. Okay, so I decided to change a little bit the title of my talk, because we have new developments and I wanted to talk about them here. So the introduction is not necessary in front of this audience, but still, just to start from somewhere, let me say that as all we know, the brain of mammals, of humans in particular, is always in a continuous state of activity. We have activity going on, and here's an fMRI that we have seen in some of the previous talks. And my goal, the thing I like, one of the things I like in this field is trying to connect structure with correlations, with function, with dynamics. I mean, with function. And for that, you need this function with function f. So you have to run some dynamical process on top of the structure that you measure to reproduce the activity that you actually see. And I became actually interested in this field already a few years ago with this seminal work of Plant's group, where they look at individual, at neurons in some slices or in vivo, and they detected these spontaneous bursts of activity that are scale-free. So they have these scale-free avalanches, power law distributed avalanches, and they made this hypothesis that the dynamics going on there could have something to do with a critical phenomenon. Because we know in criticality in physics, we have power laws, we have scale invariance, we have all these things. And this became popular, and actually there were many works claiming that criticality could be important because it entails collective phenomenon. It's the prototype of something collective. And you have also huge responsivity, huge dynamical range. You have a lot of scales from microscopic to macroscopic in sort of fractal structure. So this is a little bit of a propaganda, self-propaganda. I wrote a review paper 2 years ago on criticality and dynamical scaling, not only in the brain, but also in general in living systems, right? And in particular, in what respect the brain, we also, from a very theoretical physicist perspective, we wrote what theoretical physicists like about critical phenomena is writing field theories. Okay, we wrote our Landau-Ginzburg theory of cortex dynamics. I will not be talking about this because it's not my main topic, but we said that, yeah, if there is some phase transition, should be a synchronization phase transition. And we recovered critical avalanches, long-range correlation. We recovered a lot of things. So we kind of like our theory. But then you can ask very basic questions, also kind of theoretical physicists, like, can you renormalize your theory? Now that you have a theory, can you something like compute the critical exponents of the brain? Does it make any sense? Is there any universality in the sense that in different areas you have the same exponents, or that across different animals you have the same thing? Does all this make any sense? Maybe this sounds like very nerdy questions for some of you in the audience. But now I come to the details. And the details is that 1.5 years ago, there was this paper by the group of Bialex in Princeton, in which they proposed that they actually do a renormalization group approach to real neurons. And I will show you the details now. And essentially, what they follow is the renormalization group in the spirit of Kavanaugh. You have him here, in which you change from individual spins in a magnet to some mesoscopic spins. And in doing so, you can track how the distribution of these variables follows, and you can uncover the collective properties of the system. So let me go more to the details. These people, Bialek's group, study a mouse. You see the poor guy here in the center of this complicated picture, and he's put on top of something, he's walking, and actually he's in a virtual maze, in a labyrinth, in labyrinto in Italiano. So he's in this maze, and what they are recording is the hippocampus, which is responsible for the location, and you have displayed cells and so on. So they actually, what this the output of these experiments are time series like these ones. So they are actually following 1,000 individual neurons in all detail. You have here the time series of one of them, actually of 3 of them on top, okay? And you see when each one of them has some activity. So you have this time series that I call xi is the index of the neuron. You compute, you compute the average. So this is the fluctuation with respect to an average, and you can compute the metrics of variances, of covariances. And now the thing is that in the Kavanov's approach, you group together spins that are nearby, but here you do not really know which neuron is nearby which one. Or in other words, you do not exactly know which neuron is connected to which one. You don't know the synapses where they are. So what you do, is written down here, is that you take correlations between these time series as a proxy for vicinity, okay? So you do what is sketched here. You take 2 neurons that are the most correlated ones, and you put together in a super neuron, 1 plus 2, which is here. And then you go progressively, the 3 with the 4, the 5th with the 6th, and you go to the first step of coarse grading. Now you have neurons, super neurons consisting of 2. But then you iterate the same procedure, and you get larger and larger super neurons and all the details of the activity, okay? So let's say this K, capital K here, tells you the number of neurons in your super neuron, so it quantifies the degree of coarse graining that we have. Or more in detail, this look a bit technical, it's not. This is the new variable at the level 2, is just adding up the 2 microscopic variables that they are putting together with some normalization that you fix somehow. I don't enter the details. So you generate this sequence of new time series at different coarse grain scale, okay? And you can study, for example, what's the probability distribution of these values. And look just down here, is probability distribution following Bialec's work. We separate in 2 contributions. Some neurons that are silent, because sometimes they are, and the ones that are not. Okay, let's just study these 2 things separately for some reason. And here you have these 2. The first one here is the probability, the log probability of neurons to be silent or super neurons. And here, down here, you see the size of the supercluster, right? And this scales, but it's not linear. If the neurons were, or super neurons were independent, this should be linear. Instead, you have this scaling here that grows with K to some non-trivial exponent. So it's non-linear in K. So it's something anomalous. It's not what you would expect if neurons were independent, but they are not, of course. This is a distribution of activity, and this is quite remarkable. You see that at very different levels, they all collapse into a unique curve. This is typical in renormalization group, right? When you are at the critical point, by the way. And what you see is that this is non-Gaussian. It's not a Gaussian distribution, so it's non-trivial again. And from this distribution here of non-vanishing activity, you can, for example, compute the second moment, the variance. It's written here, again at different levels of coarse graining. And here you have the result. If neurons were completely random, you should have this, the central limit theorem result. If neurons were completely correlated, you would have this here. Instead, you have something in between. You have non-trivial correlations, okay? So you have an anomalous scaling. Everything is scaling. It's behaving very much like in critical points in the statistical mechanics. This look a bit more complicated. I apologize for non-physicists in the audience, but it's quite simple. Let me just mention it very briefly. So the thing is that they also, the Alex group, they also do a renormalization group a la Wilson. Let me just recall very briefly what this means. This means that you decompose the activity, the pattern, the spins that you have into the eigenmodes, and then you consider, you define new variables, just considering some modes. So you are removing the modes with a lot of energy. The ones with a lot of fluctuations, you remove progressively. Okay, but let me tell you something which is interesting. Here you remove the modes, but what does it mean, the modes? The modes means, it's written here, is that you take the covariance matrix, so how a spin covariates with another one, you diagonalize the covariance matrix, and you see that the eigenvalues are the components of the propagator. In other words, you give me, for each K, for each mode, for each wave plane, you have an eigenvalue corresponding to it. Okay, or in other words, the propagator, this thing here, diagonalizes the covariance matrix. But this is very important. Let me get again to the example of statistical mechanics. If you have, for example, a magnet, the easy model, you have this phase, which is order. You have a dominant mode. It's here, the origin, everything is essentially homogeneous with some fluctuations that go to zero. This is the disorder phase. Again, you have, here you have all the modes because you have noise of all possible scales. It's just white noise, right? But instead, when you are right at the critical point, you have this fractal-like structure in the spins. You have clusters of all sizes. And when you compute this propagator, you see this scale-free. I mean, this is a power law. You have all the modes nested in a very peculiar way with the power law, right? So here you see the analogous thing, but for the neurons. And what you see here is that if you compute this, I mean, let me go step by step. You compute the covariance matrix, then you diagonalize, and you see the same thing, that it's a power law. And of course, only one and a half decades, because you only have 1,000 neurons, and you have a cutoff. But importantly, this cutoff obeys finite size scaling, so you can rescale different levels of coarse graining, and you always get the same type of curve. And this is very important. So this means that this power law means that you have all possible levels of fluctuations, of variations. And the authors repeat, I skipped this part of the slide. The authors repeat the experiment for 3 different mice, and they see that this exponent seems to be, all the exponents seems to be quite robust. So I defined this one for the p silence, this one for the, I mean, the set of exponents I said before. But let me focus in this mu, which is the one of the spectra of the covariance matrix. By the way, I recommend you this very nice paper, also by the group of Bialek, called PCI meets RG. And this makes emphasis in the fact that when you have your covariance matrix, you diagonalize, and when you have this property, so that the eigenvalues go like a power law, you order them somehow with the rank, as I did before, so they follow a power law, then what you do usually in principal component analysis doesn't work. In principal component analysis, you look at the eigenvalues of the covariance matrix, and you see, oh, mostly all the variance is concentrated in one direction, or maybe in 2 directions, or in 3. And the rest of the directions I remove, and I have some dimensionality reduction, and I work with this simple, I can construct a simple model with less dimensions. But here you cannot do that, because the eigenvalues are distributed as a power law. So you have a tail, you have many of them, and in the tail you have a lot of important information, so you just cannot cut, as you usually do in PCA. You have to do something more clever, and that's the normalization group. You sum them together somehow. Okay, so in summary, we have this very nice RG approach by the group of Bialek, in which you can actually deal with real data and explore whether there is something similar. There is a scaling, there are fixed points, you have invariance of scale invariance, etc., etc. So in the rest of the talk, I will be briefly presenting 3 different things. First, I'll show you an enigma, a conundrum, and try to solve it. Then I will explore the question of universality, because all the results before were for hippocampus, how these change when you go to different parts of the brain. And then we will provide, if I have some time, some theoretical understanding. So first of all, I start with this conundrum, as I said before. And this comes from another very recent paper, a Nature paper in year 19, 1.5 years ago, 1.5 years ago. And this is by the group of Kenneth Harris in London. And they are measuring again a mouse, but now they are looking at the visual cortex, not hippocampus, visual cortex. And they look at the activity of now, instead of 1,000, they have 10,000. They have the world record in measuring the activity of individual neurons. And what they see is very similar to what we saw before. They analyze the covariance matrix, they diagonalize it to look for the eigenmodes, and they see the spectrum, and it's a power law. And they write in the abstract of the paper that this is very remarkable, that they have a covariance matrix, that when renormalized, that when diagonalized, sorry, it's the power law. And the power law is about 1 over n, over the ranking. So it's decreasing like 1.0 something, okay? This is remarkable. And they do very different recordings in different places, and it's very universal. It's working very fine. But what is even more important, in this paper, the authors prove mathematically that if this visual cortex is doing a representation, it's a neural code representing a set of images that are being shown to the mouse, then for the representation to make sense, to be continuous and differentiable, and to be operative, let's say, the exponent has to be about 1 or a little bit larger than 1, but not smaller, okay? In particular, they show different types of images to the mouse, and if you show some real world image, like here you have this exponent that I mentioned before, and if you show something which is simpler, for example, these images, then the exponent is larger. But in all cases, you have the spectrum has an exponent which is always larger than 1, okay? And changes with the complexity. So, so we said, okay, we have a lot of data. Let's do the RG analysis. Let's see how it works. And in particular, we reproduce the scaling of Bialek very nicely. This is the one for the variance of the activity. This is the one for the probability of being silent. Look at this perfect, wonderful scaling. And this is the distribution of activity. The curves are collapsing very nicely, and it's non-Gaussian, so it's not a Gaussian fixed point. And this is the one of the spectrum of the covariance matrix. Again, we reproduce this scaling, but the exponent is, as argued by Kenneth Harris, is larger than 1, is 1.1. But compare this with the Bialek's result. There we have 0.70 something. So everything is kind of universal. All the exponents are kind of the same, but not this one. This one is changing. And Harris is telling you this has to be larger than 1 for information to be meaningful. And in Bialek's, we get something like 0.7. So what's the difference? This is the enigma. What's the difference? The thing is that, well, first of all, I show you this also for, we did the analysis also for low dimensional inputs. And again, we reproduce the results by Harris. So the question is, how come that the mu in Bialek's is smaller than 1? So does it mean that it's not representing meaningful information? Okay, one is for the hippocampus, one is for the visual cortex. But what's the point? What's the difference? The point is that the data in Harris' experiment has been cleaned. So noise has been removed using what's called the cross-validated PCA analysis. I don't go to the details here, but essentially what they do is that they repeat the experiment twice. Okay, but they repeat this experiment twice and look at the covariant matrices, they're able to see what's common and what's not, and from there filter what's the correlation, the covariance coming from the input. And they present just the results for the input. Okay, so now what we did is we took the raw data, we look in the databases, and we were able to find the raw data without filtering anything, so a priori. And there we reproduced incredibly well all the results by Bialek. So even now, the spectrum, the other ones are still okay, but also the spectrum of the covariance matrix is now very similar to Bialek's, 0.72, and before it was 71, 73. So we have real universality across regions. We were also able to filter away the input. So we just focus on the noise part, on what's not related to input. And we observed, again, kind of universal behavior, but here the exponent is still smaller. Maybe you are getting lost with all these exponents for the covariance matrix. Here is a summary. So in vertical, changing the input complexity, we see that this exponent is reducing. And we have a lower limit. It cannot become smaller than 1, okay? And this is the activity associated to the input, so the neural representation. Instead, if you look at the full data, the typical exponent is this one. This is the universal exponent. And if you filter away the input, you get this, which is just controlled by noise. And this, of course, is white noise is mu equals 0. So you have this hierarchy of mu telling you what type of activity you are measuring, okay? So finally, we decided to be more ambitious and look for universality across brain regions in the mouse. And we were looking for different databases, like this one in the Allen Institute, and found data in which we have enough neurons, individual neurons, to do this procedure of renormalization. And we have these amazing results. I think it's amazing. Here we have 4 regions, but we have more than that. For example, primary motor area, auditory system, olfactive, whatever. And for all of them, we get results that are quite compatible with the renormalization group results. And as you can see here. So, and this is a matrix of the spectrum of the covariance matrix. It's not really universal. For example, here we have 0.70 something like in Bialek, 0.8, 0.6, 0.96. I mean, there is some dispersion in the values, but all of them scale and all of them have these very nice features. Okay, so can we make sense of all these results I presented? Do we have any theoretical understanding of what's going on here? So let me show you 2 preliminary things. The first is a paper by my friends in Padua, the group of Samir Suez and Amos Maritain. And they decided, when they saw the paper by Bialek, they said, okay, we do the same thing with a model that we control, not the easy model that is very simple, but we take something with some dynamics, and they took the contact process, which is a non-equilibrium system with a phase transition. They added some random noise, so it doesn't get into the absorbing phase, it's not quiescent. And they repeat the same analysis and look at this scaling, it's pretty remarkable. The numbers are not exactly the same as in Bialek, but they have scaling when they are working at the critical point. But they also emphasize, as written here in this bluish box, that the method also recovers a kind of scaling variance when you are not critical. Even in the supercritical regime, they get similar results. So this might suggest that maybe criticality is not that important, that there is something else. As I started at the beginning in my introduction, talking about our own Landau-Ginzburg theory, we also did simulations, not theoretical analysis, but simulations of our model and the lattice, and we analyzed the results with the renormalization group approach. And again, for some exponents, like this one here of the variance, we got very nice non-trivial scaling. And for others, it was not so nice, and it was not completely clear how far from the critical point you could go. So this is all preliminary. Actually, this is not a real paper, it's just preliminary results. But let me just finally, to end my talk, discuss this paper published few weeks ago, 6 weeks ago, by the group of Ilya Nemenman. And they have something which is quite remarkable, I would say. I don't understand it very well, but it's quite remarkable. What they do is that they take something like an easy model, but spins are uncoupled. So it's individual isolated spins. So how can they become correlated? They become correlated because they put some external input, some magnetic field, which is seen collectively by many of them. So it's a common shared field. It's what they call here a latent dynamical variable. It's just a magnetic field that affects some of the spins randomly, and it changes in time. So they put a number of these latent fields. They let the spins follow these fields. They become sometimes correlated because this external field, and then the results are amazing, are simply amazing. These in bluish are the results by Bialek, and this is the result by this incredibly simple model. And essentially, it's the same. Within error bars, is the same thing. Same scaling, same exponents, and same collapse of the activity. So everything works very nicely. But in this model, there is no criticality. All the correlations are coming from these shared inputs that are commonly seen for the different neurons. Okay, so what's the conclusion? Well, this is just using different parameter values. They see that the exponents are not really universal. They have some dispersion, but okay, this is a detail. So my take-home message and question, I have more questions than the final answers, is I'm gonna read what's written here. Despite of recent progress, understanding the spontaneous activity of the brain remains a very challenging task. We agree on that. Discriminating background or noise activity from input-related activity is crucial, and we are not sure how to do that. And we observe this very beautiful scaling in the spectra of covariances. This means that we have correlations nested in a fractal way in all possible scales, and then you can apply renormalization group, and that there is a remarkable universality across brain regions. So the same type of things are going on in many different places, despite that they are doing very different things. And we don't know if this has anything to do with criticality, as the criticality hypothesis suggested, or it's a mere question, it's a mere consequence of the superposition of hidden inputs of very different natures. So I think that's all. Thank you very much for your attention. I apologize because I spoke very quickly. And I have to especially thank Guillermo Barrios Morales, who has done an amazing work in working with all these databases and analyzing all these results, also with the help of Serena, who is now in Columbia University, and also my previous collaborators in the Ginsburg rundown theory. And that's all. I stop here. Thank you very much.
Thank you, Miguel. Very stimulating. Luciano, is a question? The microphone is here.
Hello. Ciao, Miguel.
Ciao, Luciano.
Your talk was fantastic.
Oh, thank you.
Congratulations. I really enjoyed it every minute. Just a little comment. Well, first, yeah, it opens a lot of clear questions. And maybe I want to comment a little bit on scaling versus universality. You know, this is not so simple if the system is not in equilibrium or if it is not self-averaging. So if you have a complex system like fractal growth that we studied long ago, so together, the universality tends to be less. For example, I think in economics, you know, this fat tails, people, some, how to say, too orthodox statistical physicists, they claim that the fat tail of economic price fluctuation should have just one exponent. I tend not to agree. Namely, let me make a little metaphor. All trees are fractal in some sense, but their exponent is different. So I would like to consider non-universality not as a problem, but as a resource. For example, biodiversity has to do with non-universality. So I would suggest to take that as a new information and not as a limitation of the quality of the analysis. Okay, just.
I agree with you. I agree with you, Luciano. It's not that I desperately want to have a fixed point with universality all across the neurons and across the brain areas. I think it's much richer in this way, but it's remarkable the degree of universality that we observe with this analysis. So yeah, diversity is good.
So there is time for another question by Maurizio.
Hi, really beautiful talk. I enjoyed to see you. And I'm wondering whether another source of elements that should be considered, not only due to the fact that these are open systems, but also that these are heterogeneous systems. I mean, you can imagine that if you have, even in the same cortical area, different time scales, so different cell assemblies that have different correlation, autocorrelation functions.
Right.
You could have a merge of them, and then you want to look at them as a whole. Maybe you are averaging time scales, and this could also produce some scale invariance, but which has nothing to do with criticality. And so I'm wondering whether this should take into account in some way when one talk about this kind of system, so heterogeneity.
Right. I don't see if you can see my screen now.
Yes, we see.
Yeah, this is the distribution of time of spike intervals in the different, one of the parts of the brain that we studied. And you see that some neurons, like this one I'm pointing here, is very localized. So it's firing very regularly. But some others at this violet here, it's firing with within 4 orders of magnitude distribution of spikes. So it's very variable. And the different colors are for different neurons. So yes, actually we see what you are saying. We see a lot of heterogeneity. also here autocorrelation function is very heterogeneous. So, but these analyses done here are kind of blind to all that. And so you just fix some time scale, which is a very important problem, how to do that. And we could discuss very lengthily about that. But the thing is that once you have fixed time scale, you see these effects. I fully agree with you that the convolution of different time scales can lead you to something that looks critical because you have many time scales in criticality. So yeah, this is a very deep and important questions. And no, I don't have a clean answer to what's going on with all these time scales.
Okay, we can thank again Miguel